Okay, good afternoon, everyone, and welcome to this webinar, EPNOE Webinar. I was already kindly introduced by Professor Xu, and the topic of my talk is about polysaccharide-based materials for human health applications. And I would like to start this with an invitation to you all to check our recently published roadmap. We have published the EPNOE Research Roadmap 2040 in open access and carbohydrate polymers. And there you are going to find very nice information about future applications of polysaccharides, challenges, trends, and so on, covering materials and engineering, food and nutrition, education part, the part of chemistry, biology, and physics. So don't miss that because it is free. So it is available for free on internet. But it's a big work with a lot of many scientists around Europe. Okay. So I work for KU Leuven. KU Leuven is located here in Leuven, right? But we have several campi in the Flemish part of Belgium, right? I'm located in Leuven. Leuven is close to Brussels. It's all about 20 minutes by train, and about 2 hours by train from Paris. Leuven is considered already for several years the most innovative university in Europe and is the 7th in the world. So what I'm leading here, I lead a group, and my work is in chemical engineering for health and care. And we have 3 focus areas: personal care, functional therapeutics, and functional prevention. In personal care, we are working with cosmetics and hygiene applications. In functional therapeutics, we are working applications for pharmaceuticals and for recently emerging cell therapies. In functional prevention, we are working with nutrition and active materials. I'm going to give examples in my talk. And we have a laboratory that is called Topochemical Engineering and Biointerfaces, or TEOS Lab. I'm also a member from several institutes here in Leuven, like this Leuven Institute for Macro Nanoscale Integration, Institute for Single Cell Omics, Leuven Brain Institute, and of course, I'm working directly with Miriam and all the executive board in EPNOE. So when we talk about human health, we are obviously need to consider the human body, right? And this is a slide I got from my colleagues here working at the biomedical department. And here, what we see is that we have the body as an organism level. Then we have the body on a system level. Then in the system level, we have to consider the organs, right? The different organs. For example, the system level, we consider, for example, the circulatory systems, the respiratory systems, neurological systems, and so on, digestive systems, right? But then we have the organ levels, right? And this complexity also goes to the tissue level. Then we have cellular level, and then we have here the chemical level. Right? The chemical here is a chemical or macromolecular level, right? When we are producing materials or when we are dealing with materials, most of the time we're working on this chemical to structural levels. And the complexity in the body is actually enormous, right? I mean, when we go here, we are mainly talking about, for example, interaction with cells, but to translate this interaction to cells, to the interaction with tissues, and from the interaction with tissues to organ levels, this is extremely complex. And this requires really interdisciplinary support and interdisciplinary collaboration, particularly because today, most of the systems that we have applied here at tissue and organ level are really, for example, plastics, right? If you talk to the doctors, if I go and talk to the doctors here at the hospital, most of the time they are working with polypropylene, polyethylene, polyurethane, so systems that are synthetic because they are inert and they provide what they need, these mechanical properties, right? So we still have a long way to go in order to be able to produce systems that will be adequate to the human body, right? We also have to consider other complexities, so other sources of complexity. For example, in the human body, we have about 37.2 trillion cells, and these cells are actually produced for about 21,000 genes, right? And these cells can be, for example, the red blood cells or platelets or fat cells or muscle cells. And we have about 21,000 genes, right, in these 37.2 trillion cells. But we have to consider that we are not alone in our body, not only with the cells, but we also have a microbiome. And this microbiome, we are talking about a multitude of microorganisms with more than 3 million genes. And we are talking about here very large, we're talking about 40 trillion microorganisms in our body. So it's about one cell, one microorganism for every cell, right? So this microbiome living in different parts of the body, either on the skin or in our intestines or stomach, mouth, and so on, they play a very big contribution. That's why we can become sick. For example, if we have dysbiosis, the dysbiosis is caused by an imbalance of this microbiome. So when we are talking about any production of materials or systems that are going to be interacting with the body, we have to consider these cells. We have to consider the complexity, cell to tissue, tissue to organ, and organ to system, system to organism. But also we have to consider the microbiome, right? That the materials that we are using are not affecting this microbiome or interacting in a positive way with the microbiome. So we also have a very complex extracellular matrix. So the extracellular matrix provides many biological clues that's going to direct cell behavior and fate. And also it is continuously remodeled to regulate its behavior. So that's why we have to be very careful when we say that, okay, I use chitosan and chitosan mimics the extracellular matrix. This is over exaggeration to say that any polysaccharide can actually mimic the ECM, right? Why? Because here if we look, for example, we have this in the extracellular matrix, we have this interstitial matrices that contains different components, different types of proteins, for example, fibronectin or elastin, collagen fibrils. The interaction between these components are very dynamic, right? In between here, the two cell membranes, right? Also, we have these hemidemosomes with a sort of intermediate filaments here in between the cells. And also, we have this literally cellular matrix, right? Where we have here a combination of collagen, 4 network, laminin, periclan, integrin. So you see here a lot of names of proteins, and sometimes these proteins are glycosylated. That just makes the system even more complex. So this is just here a note that we have complexity already on level of extracellular matrix. So this said, now I would like to take the presentation and start by thanking people, because usually you do the thanking at the end of the presentation, but I would like to thank the people and the opportunities and the funders now, because in order to tackle all these challenges, you need a lot of collaboration. And here I would like to thank my colleagues from pharmaceutical science, Professor Kivan de Motor, for food science, Christophe Cortin, Professor Gert Kamelita and Steve Stiering for the bone tissue engineering, Hans van Oostenrijk for part of biomechanics, Irene Taurino for the work of biosensors, Ruth Cardinaus for the work with soft matter, my international colleagues, Professor Elizabeth Frolini, the area of chemistry of biomass, Professor Maria Helena Melo Lima from the School of Nurseries from Unicamp in Brazil in the area of wound healing, Professor Uma Tirushi Parali from the India for the help and all the support in both in wound healing and also bone tissue engineering, and the researchers, Dr. Julie Prasad Ramanatana, Dr. Fanchier, Dr. Peter De Wever, Dr. Poonam Trivedi, Dr. Yanitrig, Dr. Armand Shobanya for all the results and all the efforts they provided to the work that we are doing. And now with the research that I'm going to present today with the doctoral students from Xiaodan King and Flavia Sancheta. So polysaccharides are life. Even with all complexity we see in the body, polysaccharides are also produced by life, a different life living organism, and they have been together at the same time as the human body. They have been tailored by nature for millions of years. So the structure of the polysaccharides, we have the chemical structure of the polysaccharides, but we also have like physical structures, physical assemblies that are formed by this polysaccharide. For example, here there's polysaccharide serving here as a skeleton for the crab, or systems like we have in lignocellulosics, right? Like the fibers giving here the strength of the wood or for the plants. So they are very there's a combination of their chemical structure and a physical structure, and a physical structure that has a function, right? The structural diversity of polysaccharides is remarkable. And what is also remarkable is that polysaccharides are also safe for humans. Most of cases, the polysaccharides are safe. And here we have nice opportunities. For example, when we have the linear cellulose, you see cellulose polymer, or chitosan, for example, here with the presence of our amino groups. So we have a polysaccharide with amino groups, or hyaluronic acid, for example, or amylose, linear amylose, and branched amylopectins, for example, present on starch, or like the very beautiful alginate, right? The alginate here with the different combinations of alpha-L-glucuronic and D-manuronic acid, and just by changing here the proportion of this, we can change the gel properties of these polysaccharides very nicely. Also for beta-cyclodextrins, that are very interesting structures, for example, for applications in drug delivery. What is interesting about polysaccharides is we have the chemical structure, right? And we can actually do this, as I told you before, this physical structure. So we can structure polysaccharides in form of gels, for example. We can make hydrogels, we can make microspheres, we can make sponges, we can make 3D printed constructs with these systems. We can do composites, we can produce films and membranes, and also fibers and meshes. And these have very interesting applications in human health. For example, in drug delivery, for example, in skin and wound healing, in applications like cell and gene therapy, in cartilage engineering, in nervous system engineering, bone tissue engineering, and also in vascular engineering. So these systems are quite interesting these structures are quite interesting for application in this in these different areas of human health, and that's why I'm going to explore today with you. So I plan now during the next minutes to focus on to present you 5 topics that has been the topic of my research here in the latest years of the team working with me. I'm just the person talking about the work of many people. So the first one is about the gastrointestinal drug delivery, and I'm going to talk about prebiotics. Then I'm going to talk about wood healing applications, then bone tissue engineering, and I will finalize with wearable sensors. So the first then about drug delivery. So here I'm very excited about a system we have that are porous microspheres. And these porous microspheres, they have a double function. They can enhance the bioavailability of pharmaceuticals because they do what is called amorphization of a hydrophobic pharmaceutical. This enhances the bioavailability of that pharmaceutical. And then we can also, at the same time, add pH stimuli responsive properties to this drug carrier. So we can do like 2 things with one material. Is the amorphization, that is quite interesting because it enhances bioavailability. And another one is to give this stimuli responsive properties. So usually there are different ways to do the administration of drugs. You can give it via injection, you can give it sublingual buccal, you can give it transdermal, you can give it topical, but oral is usually the most preferred one because there is a dosage flexibility, very good patient's compliance, few infections and contamination, and also allows your extensive drug absorption area in the gastrointestinal tract. So for this purpose, application of polysaccharides are quite interesting. Today, we already use, or if you have a tablet, that tablet contains microcrystalline cellulose, right? A microcrystalline cellulose is like a powder, right? Produced, but that powder is like a carrier. It's a passive carrier. Most of the time, the stimuli responsiveness when you use microcrystalline cellulose is coming from the coating. You have to coat a tablet to do that, right? But here, what we propose is actually to use biopolymers, polysaccharides. It can be also microbial polysaccharides, so like pullulan, for example, or dextran or bacterial cellulose, right? And we produce gels. And these gels, they can be like aerogel, they can be a gel, or they can be a cryogel. The gel production is that we usually have the biopolymer as a powder. We do a gelation stage. We have this suspension, we have this aging, and then depending the way we dry, we have a specific term for that gel. If you do this like air drying, we have what is called aerogel. So this system is very compact. The porosity is very low, but it's very interesting to exploit this to do immobilization of systems on the surface of these gels. There is then the freeze dry. The freeze dry, we can produce what is called cryogel, and these cryogels, they have pores, and those pores can be further exploited for encapsulation of drugs and so on. And then finally, if you do supercritical drying, we can produce this biopolymeric aerogel that are systems with a very, very fine structure and very high surface area. So we prepare microspheres. So in my lab here, we have systems to prepare microspheres that are simply like emulsification. We can use aqueous phase, oil phase emulsification, for example, to produce particles. And then we do this when we are producing small particles, from nanoparticles to submicron particles, or we can have drop formation techniques like this, just dropping systems, or a system using here a vibrator. Depending on the viscosity of the solution, we can use a vibrator, or we can have a system here with a voltage difference, or if we have a system with high viscosity, we make a jet and we cut this using a jet cutter in order to obtain the particle. So that's why I was telling you about a very interesting alternative of a porous system. So we have these mesoporous cellulose beads. Here are a picture of these beads produced and freeze dried. We call them cryogels, right? And they are quite interesting because they have a very interesting spherical shape that improves the flowability into the pharmaceutical sector. They allow modification. They have a high surface area and pore volume. So we have systems today that we produce cryogels with a surface area and pore volume similar as aerogels. They are hydrophilic and they have this mesoporous structure. The mesoporous structure is interesting because, as I'm going to show you, it allows us to crystallize drugs in amorphous form, enhancing the bioavailability of these drugs. So with this, we increase the solubility and release rate of poorly soluble, water-soluble drugs. We can also tailor it for a specific site delivery and release. Industry-wise, this is quite interesting because this system debates can be used, for example, in fluidized bed coating, and we can even add more functionalization or more tailoring of the systems by doing here the coating. Or we can do modifications. We can add anionic charge, cationic charge, or produce zwitterionic systems that is going to enhance either the encapsulation of drug with different charge or stimulant responsiveness, depending on our target. So why this amorphization is so interesting? Because about 60–70% of the drugs have poor solubility in water. That means they have poor bioavailability, and this is a hurdle for oral delivery. So the pharmaceutical companies, they have big list of pharmaceuticals that are very hydrophobic, but they cannot be used in formulations because of their hydrophobicity. So we have a very interest in promote the use of these cellulose beads because they can be used for adsorption of the drugs in amorphous state. Here we have to control the pore diameter of the beads in a few nanometers, and we allow here a high loading degree. So what happens is that when we have this drug that is in crystalline form, we then dissolve this in an organic solvent, and then we put this into a confined space. We are able to crystallize the drug in amorphous form. So this is more bioavailable. This can be done with silica. Silica, you can very well define the pore structure here, but silica is expensive. We have issues regarding the conversion of the system, processing of the system with abrasion, and so on. And cellulose is a much cheaper and easier solution to process. So what we do is that we take these beads, we put the drug in an organic solvent, we have it loading, and here we have our drug-loaded beads. And here we take an example, like with indomethacin. Indomethacin is a drug that when it is in yellow color, indicates that the drug is encapsulated in amorphous state. So here we see that the yellow beads here, the indomethacin was encapsulated in amorphous state. And we can confirm this by using differential scanning calorimetry and also XRD, X-ray diffraction. So this shows that the beads are able to encapsulate the drug in crystalline form, in amorphous form, so enhance the bioavailability. But our goal is also, we are able to do this amorphization, but our goal is also to add stimuli responsive properties to these mesoporous beads. And here I'm going to show you a very simple example, right? You can apply other chemistries also, but it is just like to illustrate to you that is the introduction. So we prepare these beads, these mesoporous beads, then we just do a tempo oxidation. With the tempo oxidation, we play here with different concentrations of chlorides, right? We keep the concentration of tempo and hypochlorite, and we play here with the different concentrations of chloride. And with this, we increase the extent of oxidation of the beads. And in that case, we are increasing the amount of carboxyl groups added to the beads. And here we see the controlled release of indomethacin. Here, the green curve, we see just the crystalline drug. This is the crystalline drug, crystalline that is not bioavailable in crystalline, but we can put the release this in standard body fluid. We see that here at the pH 1.2, we already have a release of the drug, right? And then when you put the pH 7, of course, the release is going to increase. Here, when we have in our cellulose beads, and particularly in highly oxidized cellulose beads, we see that we don't have the release of the drug in low pH. The pH is very low here, the release is pH 1.2, but we have the release at the pH 7. So what is the message here? This means that if we want to use this system to have a drug that is going to be taken and is going to bypass the stomach, but is going to be released only in the intestine, we can use this system, right? So as a way to protect the drug from releasing the stomach and releasing the intestine, with an advantage that the drug is in amorphous form. So this is a very simple example of the mesoporous systems. Other example we have is the opposite. Here, what we do is actually we do a oxidation with the periodic oxidation of the cellulose beads. Instead of producing carboxyl, we have here aldehyde groups, and then we do this shift-based coupling, right, with ethylene diamine. And then our interest here is to add amino groups and to add different amounts of amino groups. So we just play here with the ratio amine to aldehyde, right? We increase the ratio amine to aldehyde, and we add different amounts of amino groups. So what is the purpose here? The purpose here is to do the opposite as we did before. As we have an amino group in acidic medium, this system is going to be protonated, right? And in that case, you are going to have a system that when you take it orally, it's going to be in the stomach, and in the stomach, the system is going to release your drug while keeping the supersaturation. But what is interesting here is that the drug is going to be released in an amorphous form. Okay, so you are able to encapsulate the drug in an amorphous form and then release it into the stomach into amorphous form. Here we see the beads that were modified with ethylene diamine. We see here up to 20 minutes at pH 7. We just here moderate swelling, while here at the pH 2 to simulate stomach conditions, we see here that the beads are swelling much more, and sometimes the beads they start to disintegrate at this pH, but they keep the controlled release and they keep the supersaturation. So here we see what I told you before about, here is the indomethacin and this is itraconazole, two different drugs. So indomethacin, this is the drug crystals, and here is the drug encapsulated into the beads. You see that the drug is encapsulated in amorphous form, and this is also confirmed by the SC here. Also, for the itraconazole, the drug is also encapsulated in amorphous form, and it is confirmed with the DSC. When we look at the dissolution of the system and the effect of release, so here is the drug incorporated into the dealdehyde bead, and here is the crystalline drug. You see that the percentage of release after here or 180 minutes, you see that is below than 20%. But when we increase the addition of amino groups here, we are able to see that we can reach, for example, 9%, 90% of the release after the release time here, about 190 minutes. And this is, again, we are releasing here the amorphous drug. Good. So this is a first example. Also, we see that the size of the bead. So here we have a bead with a profile. This bead here there's larger beads, about 1.6 millimeters, and this one here is about 1 millimeter in size. We see that the smaller beads also allow here the release of the drug here about 60 minutes. You see that the smaller ones allows here a higher release than the beads that are bigger. And we see that this is preferably happening here at the pH 2. Good. So now we're going to the second topic that's about prebiotics. So, and the idea here is to enhance the bioproduction, the production of polysaccharide-based prebiotics, and this is done by immobilization of xylanase in cellulose aerogels. This is the work of our postdoc, Pedro Fardim. And here the idea is that there is a connection between the brain and the gut, right? And this connection is usually, we have the production of these short chain fatty acids, right? And the microbiota here in our gut is actually creating or producing these short chain fatty acids. And these systems here are able to stimulate production of certain immune cells, and also to regulate these immune responses. And these xyloligosaccharides, prebiotics, they are usually up to 20 xylose units with DP 2 to 6. They boost proliferation and activity of beneficial communities. They mediate levels of oxygen reactive species. They are also used in substrate and fermentative production of short-chain fatty acids. They also can be produced currently produced using acid hydrolysis. And there are novel methods here that is needed to produce them using enzymatic hydrolysis. So here we have a work where we used the beads or the cellulose microspheres. We did cellulose. We use the cellulose here to produce the microspheres, first with the pretreatment, and then after the pretreatment, we did the spirulate oxidation, and we added amines, and then we added these different types of space groups, groups that are in between the surface of the bead and the immobilization of the enzyme. So these groups are with different functionality here. The aim of this addition was to add groups that would create different microenvironments for the enzyme. And here we use then glutaraldehyde and the xylanase immobilized at the pH 7. Then we study this for the hydrolysis of beechwood xylem. So the beechwood xylem would be reacting with this system. And this is a quite interesting system because this we have spheres, we can make packet bed reactors or different applications, right? So you can also, this grafting here of different tethers, how it was done. Here we have the description after the pyrrolate oxidation. We have the shift-based coupling, we have the reduction. And here are the space arms, this 4,4-methylenedianylene, the 4,4-oxydianylene, and this 4,4-sulfonyludianylene that were added, so to create these different microenvironments. And here we have pictures of these different beads, so they have different colors. And what is interesting here is that we see that the effect of xylanase immobilization on the enzymatic activity. So here we see that all three groups, they have an advantage of immobilization comparison with the free enzyme, particularly here after certain time, 80 to about 100 minutes, we have a relative activity is higher for the system that is immobilized than for the system that is free. But the main advantage here is actually that with this system, with immobilized, we can obtain xyloligosaccharides that are bigger, longer chains than with the free enzyme. So this allows us to exploit a bit further the activities of oligosaccharides containing 6 xylose units. So instead of using systems that are, for example, with xylose triose or xylose tetrose that are conventionally present by using other systems or by using the free enzyme. So our aim here is that we use a bio-based carrier, right, that is cellulose. We attach it this enzyme, and we use a bio-based system in order to produce the prebiotic. So now we are in a phase to study these prebiotics in other systems, how they could be further exploited. Then the third topic is about wound healing. And here I'm going to talk about 2 areas. One area is about biohybrid nanofibers, and another area is about biohybrid hydrogels. And the biohybrid nanofibers. So wound healing actually involves a highly organized cascade of cellular events. So first we have this inflammatory phase here, right, where a close forms and cells of inflammation debride the injured tissue here. Then we have the proliferative phase, which is called granulation phase, right, where we have this epithelialization and angiogenesis and also granulation tissue forms and the wound begins to contract. And then we have this maturation remodeling phase, where collagens here form these tight cross links, and then to other collagen, and we have their protein molecules. So in the remodeling, you have here the closing of the wound. So now I'm going to present a work by postdoc in our group that is Giri Prasad Ramanathanan. He was a fellow, Marie Curie fellow. He was with us 2 years as a Marie Curie fellow. And his work here is quite interesting because what he did is actually a combination of is electrospinning. This is called core shell electrospinning. Here in the core, he uses this poly 3-hydroxybutyric acid, right, together with arginine. And on the shell, he uses alginate and layered double hydroxide encapsulated bacitracine. So this idea here is that the arginine would act as a precursor for collagen, and this bacitracine would have an antimicrobial effect. So the concept here was actually to produce a core shell nanofiber containing these different components, where here the bacitracine will have the antimicrobial effect. Then we study also the effect of this in vivo cell adhesion and proliferation, right? And the system from the core containing arginine would allow the production of proline. And this is going to be like we studied how this would be acting as a precursor for production of collagen. And then we did some in vitro studies of this system. And here we see the effect of a study on this cell viability of these biohybrid scaffolds. We see here the combination. See, this is just the polymer and the alginate system. This is the polymer in the center, the PHB plus the arginine and the alginate on the surface. This is the polymer plus the arginine plus the alginate as a shell, plus the LDH containing the bacitracine. And we see here that the cell viability of the system, this system is much higher in all cases here, 120 hours. And we see here also the images indicating here the cell viabilities after 24 hours in cell culture in comparison with the control. We see that our system is working very nicely. Also, the healing pattern of the excision wounds was studied here. So you usually cut an incision wound in mice, 20 per 20 millimeters, and we see the evolution of this with the application of the different systems. And we see here that after 20 days, with the system containing the polymer and the arginine, and also the shell containing the alginate with LVH and bacitracine, this closing is very good. We also did the histology studies. So the studies about we use histological images comparing the control and the best fiber, and we see that there is a reduction of macrophages here and the immune cells, right, after this day 12. And also that we saw that after the day 12, the precursor arginine is going to increase here the biosynthesis of collagen. So the concept of incorporating the arginine in the core is working because we have better collagen forming. Good. We also had a collaboration with the Brazilian team from the Faculty of Nursery in Brazil. Flavia Jancheta was here with us in Belgium, and then she did some animal studies in Brazil. And here the concept is we actually use chitosan, HPMC, that is hydroxypropyl methylcellulose, and a combination with water and glycerol. This is mixed and cooled, and then we use human insulin. So we encapsulate human insulin in this hydrogel. We have 2 units per gram of insulin here in this hydrogel, and this was performed in vitro studies and then in vivo studies. And here we see that this hydrogel enhances here the cellular viability of this human keratinocyte by testing here with the MTTSA. So we see here that in comparison with the control, for example, of 72 hours, we see that the gel that we are producing containing insulin enhances here the cell viability much more than just the chitosan HPMC and the system containing the MEM medium nutrient. Also, this is quite interesting because this stimulates cell proliferation. This is a study where we actually put a layer of the gel, we put a layer of the cells, right, on the top of the gel, and then we do a scratching. So this is a scratching system, right? And then this is for the reference, the EM, and this is for the gel. And we see that with time, there is a closure. You see there is cell migration here, huh? and there is a total closure of this scratch. So with using the gel containing the insulin. And here we see the wound closure over time. It's actually here after 60 hours, the wound is completely closed here for the system containing the insulin. So this shows a quite interesting combination or synergy between the chitosan, the HPMC, and the insulin, and enhancing here the stability and bioactivity of the insulin. Also, this was done in vitro studies in diabetic mice. We see here that after 20 days, the blood glucose of the mice actually for the system containing insulin and not containing insulin is actually practically the same. So this is not causing further glucose loss of the or detrimental health effect to the mice, and also the body weight is kept constant. So this shows that the gel is safe. Also, when we look at the area of contraction wounds for this type of gel, we see that after the day 14 or day 17, there is a closure of this system containing the gel. So the gel is working very well for closing the wounds. When we look at histological sessions of the gel, we are going to see that after the day 20, we use the gel with insulin, we have epithelialization, granulization, and we even have information here of hair follicula on the system. So in comparing with other systems, with the gel containing the insulin is performing much better, even allow the creation of hair. Then the topic number 4 is about bone tissue engineering, and here is the idea of using a 3D biohybrid matrix. So here we have a mixture. This also worked from Jiri Prasad, where we use a silk fibrin, collagen, and microcrystalline cellulose, and then we do this step-by-step freezing to produce a matrix. And this matrix then is highly porous. And then we study this interaction here with different types of cells. And then we do this biomineralization to study the calcium deposition with the potential for application in bone regeneration. So how this is done? We usually mix the silk fibrin with collagen and microcrystalline cellulose with a blender, and then we do this step-by-step freeze drying. You're freezing first with one temperature and then a lower temperature and then the freeze drying to obtain here the 3D porous structure. And what we see here is that the swelling and in vitro degradation of the scaffolds. So the systems containing the silk fibrin, they swell much more. You see here, there's a very high swelling in comparison with just with the collagen and cellulose. And also the biodegradability in vitro is higher for this system after the day 21 than with the cellulose collagen. We also look at biocompatibility with this NIH3T3 and then the MG63 cell lines. And we see here that in vitro compatibility, biocompatibility here over day 1, day 3, and day 7. We see here that for the system with the cellulose collagen and silk fibrin, we clearly see here a difference in cell viability comparison with the day 1, right? Also, for the different cell viability here for the MG63, we also see here a big difference in comparison with the day 1. So we study also the cell proliferation. We did cell proliferation studies here for the fibroblast, and for the osteoblast. And we see here that for day 3, for the control and for the different gels, that the cells adhere and proliferate very well, as presented here in the pictures. Also, there is a way to determine the formation of a hydroxyapatite by activity of this enzyme called alkaline phosphatase activity. And we see here this alkaline phosphatase activity. In the case of alkaline phosphatase activity for the day 7 and day 14, we see that after 14 days there is a reduction on this alkaline phosphatase activity, because the hydroxyapatite is formed already on the first days of the process of healing. But what is interesting here is the percent of calcium deposition. We see that the calcium deposition increases here the day 14th by when we have our silk fibrin system. The percentage is much higher than at the day 7. So this means that the system is allowing the calcium deposition. Okay, and finally, to close down, I'm going to talk about wearable sensors. Is a new research area we are starting and actually already doing nice work there with the biohybrid conductive hydrogels. This is the work of Xiaoyan King. And here the idea is to combine pullulan, the polysaccharide, with PVA. Then we do freezing of these systems, and we do freeze thawing systems. So several times we create pores, and then we soak this with ions. So we, for example, sodium chloride, and we create conductive systems. So what is the idea of application of this conductive system? It's actually to make conductive hydrogels for wearable sensors, so that we can be able to monitor the movement of a person. So here we see an example. Yes, so this is an example. This was put, you see the gels were placed here at the arm of the student, and the gel containing pullulan and PVA and the conductive system was used as a system for conduction here, so to monitor the movement. And you see here a very nice response, right? So then to summarize this talk, hopefully I could convince you that polysaccharides are an interesting application. So the human body is very complex, but we can still offer some nice solutions, like for drug amorphization and the stimuli-responsive delivery. That polysaccharide microspheres can be used, for example, for enzyme mobilization for production of prebiotic. That these biohybrid nanofibers and hydrogels can be used here for applications in wound healing, successfully containing different components. We show that systems that are biohybrid for application in bone tissue engineering, and also conductive hydrogels for wearable sensors. So polysaccharides are very versatile for applications, both for gastrointestinal, for tissue engineering, and also for external applications as wound healing and wearable sensors. Okay, so I would like to thank you. I welcome you to the EPNOE social media, and I would like to welcome you to visit us also at KU Leuven. We are very open for collaboration. Thank you very much.